afternoon. I'm Ellen Siegel, Chair and Founder of Friends of Cancer Research. Welcome to the Friends of Cancer Research first ever virtual symposium. For almost 25 years, we've convened world-class conferences around tables to tackle the most important scientific and regulatory issues to better the lives of patients all over the world. Today, this is a historic event for us and really important. We also know that COVID-19 continues to cause enormous harm. We must remember that cancer is doing the same. And in fact, cancer patients are being dramatically impacted by COVID. So all of us are really being hurt by this terrible, terrible virus. We are excited about this innovative forum today, and I want to take special time and attention to thank the amazing staff of Friends for their hard work and everything they've done. We haven't missed a beat to go forward, and I want to introduce Jeff Allen, President and CEO of Friends of Cancer Research, to get on with the important forum today, and wish all of you to be safe and healthy. Thank you.
Thank you, Ellen, and thank you everyone for being with us here today. Beginning 2 years ago, Friends of Cancer Research convened stakeholders across all health sectors to review the current methods of tumor mutational burden, or TMB, how this is calculated and reported, and trying to figure out ways to create consensus solutions on how best to standardize them. The consortia, who you can see here coming up on the screen, has worked on large collaborative analytical validation studies to support a standardized method of TMB measurement and an alignment approach, which will help improve patient care through consistent TMB reporting in a clinical setting, despite differences in the testing assays used. Ultimately, this project will help ensure consistent identification of patients who are more likely to respond to immunotherapies. Today, you'll hear from the experts who pioneered this complex biomarker. But before we hear from our incredible panelists, I know many of you with us today are asking, what exactly is TMB? To answer that, we've put together a brief video to set the stage.
For years, cancer has been treated by where it's discovered in the body. But we know that cancer isn't defined or constrained by any boundaries. In order to treat it more effectively, researchers had to change their approach and think beyond borders. This led to a significant breakthrough called TMB, or tumor mutational burden, a new decision-making tool in a doctor's diagnostic toolbox that can tell them who is a good candidate for a specific treatment. TMB is a complex biomarker that is not unique to just one type of tumor. It's a whole new way for physicians to access more information about a tumor and select the best treatment for each patient. TMB is the number of mutations in a tumor's genome. If the tumor shows a large number of mutations, it's more likely a patient will respond to treatment. We now know that two tumors in different parts of the body may be treatable by the same therapy as long as they have the same quantity of mutations. By placing the tumor on a spectrum based on the number of mutations, physicians can use TMB data to select the right targeted therapy. This means more accurate diagnoses and better outcomes for patients. It's a revolutionary new tool in our fight against cancer and a whole new way to classify and help patients. When we treat tumors based on what they are instead of where they are, that's precision oncology. That's TMB.
Hi, I'm Richard Pazdur. I am the Director of the Oncology Center of Excellence at the FDA.
Hi, I'm Elizabeth Jaffee. I'm a medical oncologist, and I'm the Deputy Director of the Sidney Cancer Center at Johns Hopkins.
Hi, Eric Rubin. I'm a medical oncologist, and I oversee early clinical drug development for oncology at Merck.
From a regulatory point of view, I think this development of biomarkers, whether we're talking about biomarkers for molecular aberrations or for the immunological biomarkers, really pose some regulatory challenges because we weren't just developing a drug, we were developing a drug in combination with a device. So this really required us to really work more closely with our colleagues in the center of, in the CDRH, really to work as a team to really develop the biomarker with the drug. And I think this poses some essential issues here because really even for pharmaceutical companies, they are in the business of developing drugs, but not necessarily in developing biomarkers. So this was a period that required a lot of discussion between not only what was happening here in the FDA, but also what was happening within companies and their collaborations with the various diagnostic, in vitro diagnostic companies. I think also, probably the ultimate of this whole development of biomarkers was the tissue agnostic indications, which we've now approved 4 of these. And they really point to kind of redefining diseases in a sense. And I think it's important for us as in medical oncology to really take a look at, is this going to be the future of oncology? And I think it will be.
Dr. Pazdur said it well. I can remember many years ago when I was a fellow in oncology, and some of the early discoveries were coming up about genes that, when mutated, caused cancer, so-called oncogenes, that were present in multiple tumor types. And there were then efforts to target those mutations with drugs. The idea, was that, okay, if these are present in multiple tumor types, these drugs might be active across multiple tumor types and not just limited to one tumor type. And it's taken a few years to get to where that's become true. I think maybe we'll talk about some of the trial designs that are needed to address a tissue agnostic indication. But I think it's a culmination of efforts that relate to understanding cancer's origins, and as Dr. Pazdur said, developing paths that enable an approval of a drug and an accompanying diagnostic, and I think just an openness that innovative approach, which is again very different than the classical way of developing drugs for a specific tumor type.
It does require people to start thinking out of the box of where the future is going rather than to, tenaciously cling to the past. Change is always difficult. And I think our staff in general, though, has basically really incorporated this into their thinking and really have embraced the concept of tissue agnostic indications and really biomarkers. They make our job, and I've stated this on numerous occasions, much more easier. It's so much easier for us to approve a drug that has a 60, 70, 80% response rate than something that has a 10% response rate, and then we're arguing about the risk-benefit relationship with that drug. Many times we were arguing, does the drug work? And now we're many times asking, how fast can we get this application out? Because it really provides an important opportunity for patients to have access to really important drugs, and we want them to have important drugs.
Change is hard in all different groups. We have our own, inabilities to change. But I think what is obvious to me, and I've been doing drug development for 30 years now, taking innovative concepts into first-in-patient studies, is that the FDA, industry, biotech, and academia have really come together. The FDA, in my mind, has really changed under Rick's guidance. They're really much more nimble and open and interactive and bringing us together. As Eric had said, Rick called him and said, why aren't you doing this faster? I feel that on the academic level as well, we're able to move new concepts into patients as long as they're scientifically sound and the study designs are good. And the other piece of this is that the patient community has really been important in all of this. And I think, again, not everyone appreciated their role, even 10 or 15 or 20 years ago, but they are a critical part of what we do. And it does have to be patient-focused as we think about things. And that's what I tell my young faculty and trainees. When you're designing your study, this has to be what patients want. We have to include patients.
Yeah, I mean, I'll just comment. It is an interesting story. I think a collaborative story, with Merck and Hopkins and FDA, so that, as Dr. Jaffee mentioned, that we were approached several years ago now with the idea that first in colon cancer, because there were some of the early work with checkpoint inhibitors in just, the average colon cancer patient, there really wasn't much activity. But then there was the patient that Dr. Jaffee mentioned where there was nice activity. And I think it was people from Bert Vogelstein's lab, Luis Diaz, and others that hypothesized that certain types of colon cancer that were known to have defects in these DNA repair genes, mismatch repair genes, had very high mutation rates. And that these could result in what are called neoantigens. So these are abnormal proteins that can be recognized by the immune system, and that might make these patients good candidates for checkpoint inhibitors. And so it started off with a relatively small trial in colon cancer, and then the thought was, well, there was good results there in these what are called mismatch repair colon cancer patients. That why would this be limited to colon cancer? And so the study expanded to other tumor types. We followed up with our own basket type study again where we enrolled any patient who had this type of genetic defect. And I remember again getting a call from Dr. Pazdur, because I think we had applied for breakthrough status in the mismatch repair colon cancer, and Dr. Pazdur said, "Well, why are you just stopping with colon cancer? Why not consider a tissue agnostic one?" And so I think perhaps we should have come to that earlier, but I think again it's back to where change is difficult, right? And so I think we were following more traditional tumor-based development paths, but of course when we got the opportunity from Dr. Pazdur, we pursued it with vigor.
It's not about the clinical trial. The patients, I keep on telling people, the patients are not here to serve the clinical trial. The clinical trial is here to serve the patients that have cancer, okay? And people many times miss this issue and want to come back to the old way of thinking. We need 2 randomized studies that show a survival advantage. And I think that's important from a regulatory point of view. And we have our own bureaucracy here that has to be moved, and that's been a 20-year job of mine to move that bureaucracy. But I'm sure Eric, if you'd like to comment, and Liz have the same issues with bureaucracies that exist in a university for Liz and in a large pharmaceutical company to move a bureaucracy to take risk, for example.
Right. Well, I think it's the same thing. I mean, we're all trying to find better treatments for people with cancer. We're fortunate. I'm fortunate to have worked with a very active drug, and I think that we're of course looking for ways to accelerate having that drug approved. And again, I'm fortunate to have been part of that process at a time when there were lots of changes happening at the FDA under Rick. But I also have to credit organizations like Friends of Cancer Research that really try to pull together, find commonalities and issues that can help to overcome some of the hurdles to getting drugs approved more quickly.
If I may, breakthrough therapy really helped me, okay, move the agency, okay? And I think people forget this. There's a clinical portion to an application, but then there are multiple other factions of that or parts of that application, including chemistry, manufacturing, which could be very problematic, okay, especially for very difficult and sophisticated drugs, clinical pharmacology, statistics, toxicology people. And when something is designated as breakthrough therapy, it does bring together people in a sense of urgency.
The other one that I think was very helpful was the collaboration around efforts that ultimately enabled the approval for the TMB, tumor mutational burden high. And again, I'll credit Dr. Pazdur for this, because this was a natural follow-on to the MSI high approval. We knew that again, much of this work done at Hopkins, that there were other ways to generate high mutation rates beyond just those that you would identify through an MSI high test. And so the idea was, again, we're leaving patients behind if we don't study those patients as well, that those patients that have tumors with high mutation rates that are different mechanisms than the MSI high. And so this was a natural idea, but it's a little more complicated than MSI high, because the measuring tumor mutations is much more complicated than measuring or declaring a deficiency in MSI in the mismatch repair systems. And so one of the issues there was trying to gain alignment across the various companies, diagnostic companies, therapeutic companies, on if you were going to pursue, again, a disease-defined indication like TMB high, how would you define it? And in particular, with the tumor mutation burden rate, unlike the MSI test, this is one where it's what would be called a continuous test, right? There's a range of mutation numbers that can happen in a given patient, unlike just being either positive or negative for the MSI high. And so this created quite a bit of complexity. And it was, again, I think through efforts from FDA and friends to pull together the various stakeholders here to say, okay, let's get together and settle on how we're going to define TMB high. And that actually happened and was actually very important for us in enabling a proper study of the TMB high population, and ultimately led to the approval.
We've talked about biomarkers, single biomarkers, but I've been thinking about mutation burden as a biomarker. We've had a number of examples where that doesn't fit all patients who respond to immunotherapy, and for many different reasons. And so how do we start to think about multiple biomarkers to identify the best patient population? Since cancer is complex, as we try to move to cancers that don't naturally respond to single agent immunotherapy, it becomes even more important to think about this.
It's not only going to be, well, I think, again, you saw that in melanoma with the BRAF and MEK inhibitors, right? So I think that's a perfect example of the proof is in the pudding, so to speak. If the drug works and you see superior response rates and superior activity with the combination of drugs, and have a strong biological rationale, which that did, obviously, because you had inhibition of two different pathways. Here again, it makes everybody's life easy, okay? It's much, I keep on saying to the reviewers, much easier to approve a drug with a 70% response rate than a 20% response rate. It's obvious here.
I just want to throw one more point out here, and that is I worry because we're talking about patients who have good access to university hospitals. And that's a very small percentage of patients. And I think as we're thinking about this, and I know we're all thinking about this, it's very important to think about it. We've thought about it before, even now, as we see how COVID is affecting people who have less access to medical care at a higher rate. This has been the case for cancer as well. We really need to figure out as a society, as physicians, as people developing these drugs, how we get these drugs to underrepresented groups and study them early in those groups and give them good access to our therapies.
Yep. Friends have really helped us tremendously in bringing around the companies that have been measuring TMB, and there's a wide variety of companies that were at the table, as well as the pharmaceutical sponsors, as well as the academics, as well as the patients. So it's a multi-stakeholder process, and I really congratulate Friends on really bringing together this diverse community of stakeholders here. And I think that's the important take-home point here.
Yeah, there were several meetings, I think, that occurred that enabled getting to sort of both, we alluded to this, that there are several tests that can measure tumor mutational burden, and some of those are more accessible in certain parts of the world than others. And so it's also important to understand if you have a mutation rate of 10 on one test, how does that number relate to another test? Is it the same number? It's a different number, because there are different ways to measure it. And so, again, Friends convened several meetings to try to understand how these assays perform. They, with discussion, led to experiments that were done to enable comparison across the various tests, and then also gathered information that was available from various companies that were that with different drugs that had information and pooled it together in sort of an anonymized way that allowed a nice discussion amongst the various stakeholders to try to get to what would be, a meaningful determination of what's considered a tumor mutation burden high cutoff.
Well, since the onset of this pandemic in March, we have been very active in contacting the patient advocacy communities, patients in general. We wanted patients to feel that they were not lost in everything that is going on with COVID, And that is not to diminish the efforts of the FDA in its work on developing vaccines and therapeutics for COVID. But we have been very active not only in our outreach program with patients during this critical period of time, but also in getting drugs out to people. We are here for you, We are oncologists. We want to make sure that we have not forgotten our commitment to cancer. And that's the best thing I could do. For all patients, they don't want false hope. They want hope that is based on reality, on science, on things that work here.
I have to agree with Rick. It is a tough time for everybody, and in particular for cancer patients. I feel the community as a whole, all sectors, are working doubly hard, maybe triply hard, to make sure that we're getting drugs out to patients. We're doing telemedicine. We've changed how we do business in the hospital setting, in the clinic setting, so that it is safe for patients to come back. And that's very important.
I think that the renaissance of immuno-oncology, Liz has been doing it for a long time, but I think for many years there was a lot of skepticism, and it wasn't until the checkpoint inhibitors came along where people realized these can be very active. But I'm not sure it's the only route. It looks promising. It seems like we get better outcomes every year. We start to see response rates in phase ones that, as you said, that are 50% or higher. Those never happened in the early days of my career. We're lucky if you got a single response. And now, we're starting to see, again, very high response rates, which I think will translate into improved longer-term outcomes.
Hi, everybody. Good afternoon. I'm Sarah Karlin-Smith. I'm a reporter with the Pink Sheet at Informa, and I'm here with a great panel to expand on the conversation we just heard. I'm joined by Brian Alexander, the Chief Medical Officer of Foundation Medicine, Matthew Hellmann, a medical oncologist at Memorial Sloan Kettering, Jamie Holloway, a molecular science liaison at Caris Life Sciences, and Stephen Lemery, the acting director of the Division of Oncology at FDA. And I wanted to start with Dr. Lemery. In the earlier discussion we just heard, FDA's Rick Pazdur talked about how biomarker-driven drug development has often made FDA's job easier because the benefit of the drug is just often very clear and strong. And I wanted to get insight from you as to how else this biomarker-driven drug development and complex biomarkers has changed the work of a regulatory agency like FDA.
Well, I think in general, it's really forced both companies and FDA to think about how the drug should be developed best sooner. In essence, should the drug be developed in a biomarker-specific tumor type, all tumor types? Should they do multi-cohort trials? This is so it's led to increased thinking about the development early and nimbleness of changing the development very early. We have more complex trial designs now, which has led us to think about these more complex trial designs and what the statistical plans should be, as well as how to monitor safety in these trials, which is increasingly complex when you have trials that may be enrolling 1,000 patients across many different cohorts. I think there's challenges regarding the optimal dose of these drugs because, for example, with NTRK, we approved the trials, the drugs based on, I think, fewer than 100 patients. So it's harder to get. You can get an effective, safe and effective dose, but is it the optimal dose? And that's an additional challenge. And finally, we have to consider very early how to go about thinking about the other aspects of drug development, including manufacturing, including non-clinical development, including the biomarker itself. And sometimes these has to be thought about even at phase 1, very early in development, which, you know, in the past 10 years ago, you didn't necessarily have to do that until you're about to start the phase 3 study.
Thank you for that. And I also wanted to bring in Dr. Alexander here. Another interesting thing I thought in listening to the discussion was Merck's Eric Rubin. He discussed how important it was that a common definition of tumor mutation burden was created because these complex biomarkers are often difficult to define and measure. And I was curious, working in this space, what else do you think needs to be done to encourage harmonization across the testing industry to help players in this field?
Yeah, no, thanks for the question. You know, as the biomarkers get more complex, it's even more important to have kind of harmonization amongst different kind of organizations, because it's a communication issue, really. And you know, we work very closely with Merck and FDA to kind of be the companion diagnostic on this pan-tumor pembrolizumab approval with tumor mutational burden. But honestly, like, that's not going to be, people aren't going to internalize that as terminal, and it's not good for the community for people to internalize that as tumor mutational burden as measured by Foundation Medicine. So, you know, if we don't have harmonization, there's going to be a lot of confusion. So, you know, I think most companies have incentives to harmonize. I think that there's no winning, and it's not being patient-centric to have a proprietary way to measure something that no one else, that no one else can. And we all have an incentive that there's not some kind of rogue player that calls something tumor mutational burden that isn't, and has a bunch of failed kind of trials and creates a lot of confusion in the space. So, you know, I think being patient-centric and, you know, thinking about how we can provide the most value to the oncology community, harmonization makes sense. That's why we were participating in the Friends kind of effort. So I think it's really just like groups like Friends providing that kind of catalytic energy to bring these groups together so we can have this discussion with a patient-centric mindset and say, what is, you know, what's best for the community, and how do we play in this space?
I wanted to quickly follow up on that question because we had an audience question that relates to this. Sujen Chen from Actinomics asked, what's the next complex biomarker that needs this kind of harmonization effort? Dr. Alexander, do you want to take that, or if anybody else wants to chime in?
Sure. I wouldn't say that we're ready to move on from tumor mutational burden. I think, as you heard Dr. Rubin say, this is a continuous variable. Saying something's high or low is kind of like saying, is it cold outside, and setting a cutoff of 60 degrees and saying, should I wear a coat? If it's negative 20 degrees, yeah. If it's 59, is that different than 61? So, I mean, I think even tumor mutational burden is more complicated than we use it now. And so I think that harmonization, then, you know, PDL1 is, IHC is something that gives complementary information. So, you know, a lot of these immunobiomarkers are measuring one aspect of the immune response to the drug and the tumor. So we don't have these kind of like on-off, like, is a mutation, will respond, if not mutation, will not respond. And combining multiple different kind of dimensions of biomarker measurements into an immunoscore is probably the next really complicated step, and I think requires a lot of transparency, a lot of like, what's inside these algorithms, how do we all work together, and are we measuring similar things?
Does anybody else want to take that question of what the next complex biomarker that needs to be harmonized is, or do you all kind of agree with Dr. Alexander that the focus still needs to be on tumor mutation burden right now?
Sarah, I would say that we also need to be thinking about the HRD pathway and thinking about loss of heterozygosity and homologous recombination deficiency, because we have a few new approvals now that are looking at different aspects of that same pathway, and everybody sort of looks at them a little bit differently. So I think those are going to be some important, another important complex biomarker for us to consider as a group, thinking about how to harmonize it and how to use the same kind of nomenclature.
I completely agree with that. I don't think we need, I think we can walk and chew gum. There's a lot of the new biomarkers that are coming out are more complicated than ones in the past. And HRD is a great example of one where multiple different kinds of definitions, different approvals, different diagnostic tests. And so coming together on that as well.
Great. Well, Dr. Holloway, since you've kind of entered our discussion, I wanted to keep going with you. One of the things I thought was interesting in kind of talking to you all and preparing for this conversation, and it was also an issue that Dr. Jaffee raised in the earlier discussion, was that a lot of Americans who don't have access to academic medical centers won't actually gain access to the types of clinical trials and biomarker cancer-driven medications that we've been talking about here. Have you thought about what can be done to change that trajectory at all?
Sure. Dr. Jaffee makes an excellent point. We know that there are disparities in outcome based on where patients are treated, and certainly concern when we think about the fact that as many as 85% of patients are actually treated in the community setting. So I think it's really important that as we think about being patient-centric, that we think about how we can make sure that they have the same resources available to them as those who are treated in larger academic centers. But I think that even though complex biomarkers are by nature complicated, I think we could consider that if they're well defined, that they could present us with an opportunity to change that trajectory. Because if we're going to prioritize the harmonization and, as I said, the definitions and the nomenclature to get those all the same, that can actually reduce our reliance on these big academic medical centers to gain and understand that important information. So if we can give confidence to doctors in the community setting that results from different labs are comparable, then they can send their tissue samples or even blood out to analysis to large labs that have the technological resources, state-of-the-art facilities. They have the expertise to provide not only the details about these biomarkers, but then also to maybe provide some evidence-based insight to help doctors understand the consequences of some of these biomarkers. And then, of course, the last part of her comment was about clinical trials. And I think we all understand that just knowing that you're eligible for a clinical trial doesn't mean that you're going to be able to enroll in that clinical trial. There are lots of barriers. One of the biggest ones is always geographic. And, you know, so many of these trials are still housed in academic medical centers that the average patient doesn't really have access to. And so this is where there's a really great opportunity to work with just-in-time trials organizations that can make these trials accessible in the community setting. So they make it easier to open them quickly and efficiently, but in the local community oncology practice after the identification of an appropriate patient, so that then you're able to retain care of that patient in your home clinic, but most importantly, the patient's able to be treated in a place that they find comfortable and convenient. And so I think we're gonna have to think, like Dr. Pazdur said, about doing things a little bit differently than we're used to doing them and putting all of our resources into the big centers. But I think that there's a way to do it that can really improve access for patients who are not being treated in those very large centers.
Yeah, I might just add here, I've heard Jamie talk before about democratizing it, some of this information, and this is probably a place where the diagnostics may be farther ahead than the drug access, because when we heard Dr. Jaffee talk about access, it was more drug access than it was the actual biomarkers, and we've seen it at Foundation, and sort of the same with Caris. Most of our interactions with the community oncologists, so you know, they have a companion diagnostic TMB test that's available to them, and other biomarkers that we work hard to have companion diagnostics, and we work closely with the FDA on. So I think, you know, having that kind of validated like information is important. Then be able to use the information is another step. And then once you're able to kind of like use the information to be able to pull it through to an actual decision. So whether it's trial availability or drug availability, you know, I think we're also interested in just-in-time trials, decentralized trials, because now we have this mismatch where you can get a result anywhere that's kind of the same, you know, or better result as you get at some academic medical centers in terms of the diagnostic, but then do you have access to the drug or a clinical trial? And, you know, how do you kind of you close that loop, whether it's different kind of trial models or kind of working more closely with academic partnerships. And so I think, you know, that democratizing of the information is a step that a lot of groups have made, and then acting on that information is probably the next step that needs to happen for it to be pulled through.
Thanks, both of you. I wanted to bring Dr. Hellmann into the conversation. You're working at Memorial Sloan Kettering in clinical practice. Can you talk a little bit about how your practice on the ground has been impacted by biomarker-driven drug development? And when you look towards the future of the next maybe 3 to 5 years, how do you see it evolving even further?
It's a great question. It is fundamentally the way in which our care has changed in the last 15 years through trying to understand the genetics of cancers and to partner the best drugs to the right patient. It's with that sort of lock and key mechanism that I think we've made the most progress thus far. And in that context, this approval of pembrolizumab in patients with high TMB represents a continuation of that same progress. That through a detailed molecular assessment of a patient's tumor, we can identify the right drug for a given patient. And that way, tumor mutation burden represents sort of a part of that molecular testing that we are already doing routinely and could do more. It's information that's often gained from the same sort of molecular tests that we're doing to try and treat patients with all sorts of different cancer medicines. That's how we identify EGFR targeted therapies or ALK or RET or BRAF or ROS. These are all individual targets that if we know which a given patient has, can remarkably change both the treatment options and their outcomes. And so tumor mutation burden can be identified through similar sorts of tests. It doesn't have to be a distinct test onto itself. It's not like we have to test for TMB per se, but rather this is additional information that we can get from the same sort of molecular analysis that we've been doing. I think the addition then of pembrolizumab for patients with high tumor mutation burden then is the next step in this journey of precision immunotherapy and gives us the opportunity to use these medicines, which have been so transformative in so many different tumor types like lung cancer and melanoma and kidney cancer, but extend them to other diseases which are not routinely treated with immunotherapy. But if we knew a little bit about the molecular landscape of a given patient, we could use these medicines in an informed way to be able to get immunotherapy to patients that have uncommon tumors, but they might just have the right molecular lock and key. And that's incredibly gratifying when you're able to bring potentially life-changing therapies to patients that would never otherwise have had that opportunity. So I think that this is both a continuation of the ways by which we've made real progress in the last few decades in the treatment of patients with cancer in general through molecularly targeted therapies, but of course represents like a new step forward in that frontier of using that information to direct the use of immunotherapy as best as possible.
So we have a lot of good questions from the audience coming in, and please feel free to keep sending them, but I want to ask some of them now. Peggy Eastman of the Oncology Times asked, what level of validation is required for a biomarker to be integrated into clinical practice in oncology? I think a number of you maybe could answer that from different perspectives. Perhaps, I don't know if Dr. Lemery, you want to start from the FDA perspective of kind of how you validate biomarkers as part of the approval process.
Well, the analytical validation of biomarkers is probably beyond my expertise. That's in the domain of our device colleagues within the agency. Clearly we want the device to be able to measure what it's going to measure, and I think at least in speaking to some of the rare fusions like NTRK, it's given the high response rates in those, we're pretty confident that those tests were identifying true positives. However, clearly there's a question of like, how's the sensitivity of the test? So clearly there's the analytical validation. CDRH is also considered clinical validation to support approvals of these devices. Ultimately, I think as a patient, you want for next gen in particular a device that really is analytically valid across the entire panel rather than, a perfect test for NTRK or a perfect test for TMB, but, suboptimal for everything else is not an optimal test. You really need, as a patient, the entire panel to sort of function in an optimal way.
Dr. Hellmann, I was wondering if you wanted to jump in here as a clinician on the ground. Is there something that you look for to decide whether to incorporate a biomarker into your clinical practice?
Yeah, I think probably there's two things. One is it feasible? Is it feasible to do using the routine tools that we have available in clinic? And is it meaningful from a clinical perspective? And so in this context, I think both of those criteria were met very clearly. The feasibility is built upon the broad next generation sequencing that we are already doing for many different tumor types as a matter of routine. When we had initiated some of the research related to tumor mutation burden and response to immunotherapy, much of that was built upon whole exome sequencing, which from a methodologic and informatic perspective is probably too complex right now to be able to do at scale routinely in clinic. But when we transferred some of that information into the targeted next generation sequencing panels that are already clinically routine, we found that we could estimate tumor mutation burden with, I think, pretty good accuracy. And so that provided the feasibility that was possible to make this something that is accessible to all patients. And then the second part was whether it was clinically meaningful. And I think clearly what the data across a variety of cancers, including the data as part of this particular filing, has highlighted is that if you look at patients that have high tumor mutation burden, they reliably have some increase in response rate to pembrolizumab. And it gets to a certain threshold of which if you were to see a patient with an uncommon tumor that would not otherwise have the opportunity to try immunotherapy, I think if you pose this opportunity of saying, well, we've done this molecular testing and it seems like it increases the likelihood of benefit to a meaningful degree, I think we should give it a shot. I think most patients would say, yes, please. And so I think in that context, this has really brought a new opportunity for patients that I think is quite important.
So I have another audience question here. This one is from Elaine Shatner at Forbes. She says that she's seen a lot of criticism of TMB by some oncologists on Twitter, everybody's favorite platform, I think, for criticism and debate. And she said these oncologists claim it's not useful, and she wants to know what it will take to persuade the public and most doctors that this test and not just Foundation's brand of it is worthwhile. Perhaps that's a good one for you, Dr. Alexander, to start out with, and then if anyone else wants to jump in.
Sure. I think there's a lot there. I've seen a lot of the discussion on Twitter, and I think it brings into a lot of different kind of philosophical discussions about who's kind of making decisions and what access to therapies and those kind of things. I'd say to your last point, and not just Foundation medicine, probably circles back to the harmonization question that you had before. It's like, how do we know that we're measuring similar things with different tests? You don't, to my temperature analogy, you don't want to have to know what thermometer you're using to know that it's 60 degrees outside. To the kind of more the kind of controversial topics, I mean, I think some of it was focused on response rate as an endpoint. Some of it focused on I don't know what you use instead of that across in a pan tumor kind of approach. So overall survival is different for different therapies or for different kinds of tumors, and disease-free survival is different for different kinds of tumors. So as Steve was talking about before, clinical trial innovations are going to be important here. There's also a discussion of whether all the tumors that were in the cohort responded in the same way to make a pan tumor claim, and I think part of that is your I guess philosophical approach to whether you want decision making to be more available to doctors and patients to make more complicated trade-offs and have that information available. As biomarkers get more complicated, so is the decision making in a lot of ways. As we said, kind of TMB as a continuous variable. Maybe something where you have to think of a probability of response versus other kinds of therapies that you'd you'd have, whereas in targeted agents, it's more of a algorithm. It's like, if I have mutation A, I give drug A. If I have mutation B, drug B. And because it's kind of more binary and the responses are yes or no, it's an easier decision algorithm. Whereas once kind of the biomarkers get more complicated, it's much more going to come down to decision making between patients and physicians, taking all the information that's available, really kind of saying what the trade-offs are, what we think the next best therapy is. And so I think in that context, tumor mutation burden is hugely beneficial. If you cut it in different ways, people can have different criticisms with how something was rolled out. But I think that's why we need continued evolution of the development of these complicated biomarkers outside of just the drug development paradigm.
Does anybody else want to weigh in on that question? Dr. Lemery, I saw you sort of shaking your head a few times. Do you have any thoughts on kind of how to persuade the public and doctors about this test?
No, I mean, I think Brian did a great job, with the issues related to the approval. I think, the comment earlier also about the extension of, what we already know about the drug is a good one. The drug's approved for, the vast majority of patients it already had approvals for that have high TMB. Your patients with melanoma, your patients with lung cancer, your patients with MSI high cancers, different tumor types have, these are tumors often with higher TMB. So now you have, rare tumors, neuroendocrine tumors, for example, or sarcomas, these patients may have, a higher TMB, and it's in essence an extension of the approvals that have been given to a drug that we know, that thousands of patients hundreds of thousands probably of patients have received it. We know what the risk benefit profile is. We know, we have an understanding of what, effect on response may translate into, given the durability of some of these responses. So it's not like it's a new drug that's first time approved. It's we have such a big body of data with this drug. So I think as was said earlier, it's an extension of what we already know about the drug, I think.
Dr. Holloway, I think I'm going to throw this question to you because you have sort of experience as both patient and scientist. Ken Valkenburg of PGDX asks, with all the different biomarkers available now, is there enough effort being placed on education of patients so that they understand what's going on and what their options are?
That's a huge question. I think Brian sort of alluded to it already, that as these things get more complicated, we are placing more responsibility really on the patient to participate in these discussions that are really pretty complicated. And so I definitely think that it's incumbent on us as scientists, as the community, to make sure that we are being effective in our communication and that we're being mindful about the words that we use. That's another reason that having good nomenclature is so important. Having standard definitions between companies, just to make it really clear to patients that they're understanding the words that are being used. So if they read the label, the FDA label, that the words that are being used there are the same words that all of the companies are using. And I have a really big passion for educating patients, so I'm never probably going to say, oh, we're doing plenty, right? I'm always going to think we could do a little more. But I think even forums like this are really excellent to give patients an idea of the breadth of things that there are to understand, and to try to break down to sort of the really important things. And that the example of the Pembro approval in TMB is a great one, because like Elaine mentioned, there was so much discussion on Twitter, and a lot of the patient groups were very skeptical because there was no survival in any of the approval data. But I think it goes back to what Matt was saying. The approval was for patients in late stages. They don't have any other good therapeutic options. So if you can give them one more option that seems like it's going to have a decent likelihood of response, and as we saw in the data, there was very durable response when there was one, being able to break it down into that kind of like simple language to really help patients see the priorities and help them put it into context of their priorities, I think is important.
Okay, I want to take another question from Takeda, Christine Ward. She says TBM is a great example of immunotherapy biomarker with pan-tumor predictive value. Why haven't we seen a similar approval yet with PD-L1? Is this due to the data not being available or due to challenges during the review process? Not sure who might be best to answer this question because it mentions the review process. I don't know if Dr. Lemery, you have thoughts, or if someone else wants to take this one.
Without reviewing I haven't reviewed all the applications. I think PD-1 has been a little bit more of an inconsistent biomarker in different tumor types. So I think it's been a more challenging biomarker to sort of, to say if you're PD-1 positive and at what level that you're likely to have benefit of the drug. I think that's been one of the major issues with PD-1, especially as compared to TMB.
Okay, another questioner mentioned PD-L1 as well, and pushing us to talk outside of just tumor mutation biomarker. So they wanted to know, this was from David Rim of Yale, can we talk a little bit more about comparing the new TBM test to PD-L1 testing?
Yeah, I think there's a lot of interest in that. I think that in a lot of the, we do a lot of PD-L1 staining at Foundation. And I would say that, I think you need to think about the decisions that people are making and think about how the biomarkers impact those decisions. And, I think what we're pretty confident in is TMB and PD-L1 aren't measuring the same kind of aspect of a response. So that means if there's independent information, we have to figure out how to combine that information to say something better than either one of those alone. And then there's other kind of genomic signatures that you could probably add in there, host kind of HLA typing and types of things. So the immune biomarker is, biomarkers of response to immunotherapy are only going to get more complicated as we combine multiple dimensions. So PD-L1 is certainly, a piece of that. And I think that we've seen, we have some retrospective data with our Flatiron colleagues that show kind of independence between PD-L1 and TMB. And I think Matt has shown, Memorial has looked at that as well, and others. So, how to combine those into kind of a single score, I think are the next steps in these biomarkers.
Okay, we have a special request for Dr. Hellmann. This question is for you. And I apologize, I'm probably gonna mispronounce this person's name. Twinkle Marfatia of Personalis asks, would you prefer to order exome Y and TBM tests compared to panel-based TBM tests for your patients? And I apologize, I keep also, I think, inverting the T and the M and the B. Or do you see only cost as a barrier, or do you see any other barriers, I guess, into which panels you're ordering?
I guess it's a little bit of a tangential question about sort of molecular profiling of tumors. Undoubtedly, there's probably some additional scientific information that can be gleaned from exome-wide analyses. I think it's less certain that additional information has routine clinical value. In that context, the amount of information that we've been getting from panel-based sequencing, which provides a degree of depth and coverage that is not as routinely feasible with exome sequencing, has been really transformative in terms of the way that we examine and treat patients in general. And so, on one hand, I guess I'm wary of sort of comparing and contrasting as if they are in competition with each other. I think that they have served complementary purposes in our work thus far, with panel-based testing being feasible at scale and with depth for routine clinical use, and exome sequencing being useful for discovery and scientific pursuits. But it's important that we differentiate those two goals. And so in that way, like, could I imagine a future in which exome sequencing is routine for all patients? I think is possible. And yet I wouldn't want that to be misinterpreted as sort of fundamental flaws in panel-based sequencing, which has been, I think, really important for clinical use, and has provided, in the narrow context of the conversation today, I think has provided much better estimations of tumor mutation burden than I may have suspected from when we first began this work. By sequencing just 1% of the exome, not 1%, like a 30th of the exome, we're able to sample the mutation burden with pretty reasonable accuracy. So I think it's been a really important tool. And I have no doubt that the molecular tools and the depth with which we can examine the genetics of tumors and patients, for that matter, will undoubtedly improve over time.
Thank you, Dr. Hellmann. And this has been a really great panel. We had a lot of audience questions that were really fascinating. And I thank you all. You made my job much easier with all those interesting questions. Thanks again to all our panelists and our audience for joining us today. I want to turn it over to Jeff Allen of Friends of Cancer Research before we finish up for some concluding remarks.
Great, thank you, Sarah. Many thanks to all of you for joining us as well for our first Friends of Cancer Research virtual symposia. Please join us next week, though, on Thursday, July 23rd at noon Eastern for the second half of this discussion, which will feature the full presentation of new results from the TMB harmonization project. We definitely hope to see many of you then. Until then, stay safe and be well. Thank you.